Okay. Yeah, I think I'm recording it. Can you see it?
Yeah.
All right. Okay, so let me see.
I think new students join us now.
Yeah.
Yeah.
So hello guys. So you are interested in our research. Thank you so much for that. If you are not here in the first section, so I'm gonna start the second sections now. So let me see. My name is Hayden and my friend is Afaf here. We are work under Dr. Theravadu to do extracting and isolating anti-cancer components found in the black nightshade leaves and then the yellow turmeric roots. So as we know so far that we have several methods how to treat the cancer cells, which is biological therapy, chemotherapy is really famous now, and then radiation. So all of them, they all have the side effects to the body, to the human cells. So that's why we come up with another method that actually been using across the world, which is the herbs, which go back to a plant. They also have the anti-cancer activity within them, and they're less harmful to our body. So that we on the first steps, we extract and purify the black nightshade leaves using acetones as a solvent. So we extract the plant group and then put it on the thin layer chromatography, the small one, to see if our solvent is the best to separate the compounds within the plant. And my chosen solvents here is the petroleum ether and acetones ratio 4:1. That's actually better than the solvents that's been used before. I believe is ethyl acetate and hexanes for the 30. Since I have more than 10 different bands coming up on that TFC plate. So after that, after decided the solvent, we moved it to the silica columns chromatography, which is similar to the thin layer, just upside down. I'm going to show you in a minute. So that when we use the gravities to, and then the different in polarity to isolate the different compounds in my samples. And then we collect fractions from those steps. And then, but when I collect the fractions, each fraction actually have multiple compounds, which is not really promising that I isolate anything. So I come up with another approach, which is instead of that silica, I just load directly my samples on the large TUC plate and then let it in the chamber. When the solvent run, the compounds that have different polarity run with it. And then you can see that different bands show it up right here. So I basically just scrape them up, put in the beakers right here, and then I use the suction filtration to wash the compounds that stuck in the silica down to the filtrate, and then the silica will remain on the funnel. So then the next step would be after I filter them all, I'm going to test it with the IR and NMR to determine the functional groups as well as the structure, and then I come up with a next step is going to be testing on cancer cells. And let's see. So Afaf, can you please talk a little bit about your samples?
Yeah. So as Hayden said earlier, our objective is to extract anti-cancer component from different herbs. So in order to treat cancer, both cancer cells and cancer stem cells have to be killed. However, chemotherapy which is the most used to treat cancer, only kills the cancer cells without killing the stem cancer. So why stem cancer is that much tough to be removed? So the reason for that is there is a cytokinase which called interleukin 6. This protect cancer stem cells from medication, like for medication who destroy the DNA of cancer cells. This cytokinase protected. Therefore, there is a study shows that curcumin, which is a component of yellow turmeric, works in both killing cancer cells and stem cancer cells. So therefore, using curcumin will be much safer and more effective. So as you can see here, the first picture, so this picture after doing extraction and evaporation. So as you can see here, the solvent I found were able to separate the three components. So after that, I did the silica column to separate them. So as you can see here, the two pictures are from the 40 fraction I collected. So the disadvantage about this method, first, it takes between 6 to 8 hours to complete the process, and then it's expensive to do. Therefore, I moved to the next approach, which is using my sample directly on big silica plate. After that, I scraped each line. So each line represents one component. So I had 3 lines, which is 3 components. I separate them in different beakers. After that, I washed off the component from the silica using acetone. So the step we will do after that is to test this component on breast cancer cell. So Hayden, if you want to show them the process in detail.
Yes, my pleasure. So here, the couple of slides next gonna be individual steps that we did in the lab. Let me see. Okay, so first I want to show you the pictures of the black nightshade plant that I found. This actually on the walkway to the STB building. So it's indicate that this thing is everywhere. So we have a great medicines everywhere that we didn't really know of. So this the first step is the extraction. So I took the plant, weighed it, and then blend it with a blender mixed with acetones. So the acetone is going to break up the chemicals in it. So after that, I put it on the stirring plate to stir it 24 hours under the hood. And after that, I transfer it to a round bottom flask here. And this machine here is the rotary evaporator. It's going to take off the solvents out of that samples, right? So the acetone is going to be soaked up here, and then they have a little bit like a chamber to collect the solvents. It's connect with the condenser. So after all this, the acetone evaporated, we're gonna have up here extract right here. This look like a gel, like stick on the bottoms of the beakers here. So this is the video that I filmed it. Wide in set of extractions and concentrations here. Yeah, so you can see that this is the temperatures of acetones, boiling point of acetones. So this is the rest of the acetone that's collected here. After this, we just take it out and dump it. And then let's see. After we have this plant root here, this is ready for the silica columns. See, So this is the burette that loaded with silica and here, and then the solutions here is the chosen solvents that we use. So I use the petroleum ether and acetones ratio 4:1. And then the next picture here is when I loaded my samples in the columns. And when I turn on the knob here, the gravity is gonna let the compounds run down based on their difference in polarities. So let me show you this. This process is a long time. You can see the sky behind it is like 6 hours long. But the process looked really good. I love to see this whenever it's run down here. You have like a different, you see the different colors indicate like different compounds that run down to the test tube that I placed out here. So my plans at first, I just want to collect 40 test tubes. But since the columns still have colors, so I don't want to miss anything. So I just keep collecting and collecting whenever the silica is totally white. So that, let's see, so that I have total of 112 fractions. But you can see that they have different colors, have different compounds in it that reflects based on the colors itself. And the next step is tests on the large silica plate to see that how similar they have in each fractions. So if they have a similar polarities like this, I just combine the fraction that have the similar IF values like this. And the next pictures here is the combined fractions that I determine based on the RF values here. So you can see that this the yellow oil compounds here, it's non-polar. It's run top of everything. So like it's go down the column first, and or even on the plate, it's actually the first band on the top, which indicate that it's really non-polar here. And then they have like applied colors, a light blue, green. So I'm really happy with this result since I can I didn't really isolate, but I actually can reduce the compounds within 1 fractions. Since my fractions still have multiple compounds in it, so I decided to try another approach, which is I load my sample directly on the TOC plate. It's gonna be the same property, just like the silica columns. So when I put it in the chamber with the same solvents, still the solvents front still going up, dragging all of the compounds with it. And then after done, you can see like with the silica columns, the yellows run down first now. With this, the yellows run first on the tops. So I can actually use the flat spatula to scrape off the silica that have the bands that I wanted. Just take it off, put it in the beakers right here, and then it's ready for suction filtration to take that compound off. So this is a little video that I filmed. So you can see that they actually still overlap, but the moment it's run on top of the plate is actually isolated. So I can easily say that I can isolate the first band, this first yellow band here. There are 3 more that colorless, in the middle of these, and then the first black bands and second black bands. So this method, it actually give me like a faster approach to isolate this type of plant. All right, let's see. Okay, so Afaf, can you talk a little bit about your procedures? We actually have the same techniques, just different compounds, so it's pretty simple.
Yeah, so this is a summary of the steps I did. So the first step is extraction. For extraction, I separated the skin from the pulp. Therefore, you can see 2 separate beakers. After that, after 24 hours, I did evaporation. And then the third step is finding the right solvent. So as you can see here, the solvent I found is able to separate the three components. The fourth step is the first method for the fourth step is using the silica column. So using the silica column wasn't effective. Therefore, I moved to the next method, which is using my sample directly on the silica plate. And this method was effective. So I can see here a question from Hafsa, which is, what's the longest step in your experiment? So I would say step 3 and step 4. For step 3, it was hard to find a solvent that is able to separate the three components. And then for the fourth step, only the first method was long, but the second method wasn't long. The first method took about 6 to 8 hours, but for the second method, only, I would say 3 hours. Yeah. How about you, Hayden? Do you want to share what is the longest step?
Oh yeah, I actually have the same impression with this. So the longest one, yes, would be the finding the right solvents for your compounds. It's, let me see. I was a little bit lucky though. Like this is when luck actually come with your choices. So I first started this project in summer last year, and it took me like 2 months just to find that solvent. So like it's basically the whole summers to find that solvent. It's funny because some kind of solvents, they have really similar in the polarity index, but they actually separate your compound differently just because of that slightly different. So I can show you here is the slide that I use for finding my perfect solvents here. So these, most of these I have used it all. I have combined them with different combinations with different ratio. So I would say that if you say how many times did I try, I would say like hundreds, because it's like I really want to show you my books, like my notebook, they're really thick that I glue every single TOC plate in it. If you want, you can stop by my lab. I will show you like anytime. So yes, so in order to answer that, yes, so this step is the longest steps to me to identify the perfect solvents. And then the next longest one is the silica columns is indeed longest because it takes time for those compounds to run down. And I try many steps, many methods to try to like push them down faster. I use the air in the lab. They have like the air and the vacuums. I just plug a tubes, and then I try to like flush it down the columns to fastening the compounds. But no, it's so whenever it's go down, it's actually get closer. So it's mixed a little bit more whenever I try to speed up the process. That's why I decided to just sit and wait. And then sit and wait for 6 hours long in the lab is so bad.
Yeah.
Yeah.
Yeah. Also for the silica column, it's hard because you have to pay attention for the silica to not dry. So yeah, this also can play a factor. Yeah. I think, Hayden, you already answered next question, which is like the solvent can be in the same range, like two polar can be like in the same in the same range, but like they will not work the same with separating the components.
Right. So similar here. So you can see like down here. I picked for the polar one, I actually picked acetones for my first. And then for dows here, you can see the petroleum ether and hexanes. They are like next to each other. So I believe that they different by like 0.1 in polarity index. But when I mix them, they give me not a total different result, but with the petroleum ether, they actually separate more of my compound than the hexane that I use. So that's just very critical here for your compounds. So in order to know, you can't just tell by the polarity index. You have to try it. No, I didn't do the 1-to-1 mix. It's gonna be the first start off of everything. Just mix 1-to-1. But for my compounds, I try a different like various of ratio, like maybe like extreme, like 1-to-20. It's actually really good because everything so like 20 nonpolar, right? So anything that nonpolar is gonna go really fast, but then anything that polar is gonna stay on the bottoms. With that extreme, you can easily isolate the compounds, but it's take a long time for you to load all of that in the columns. So I wish that I had like a big column to try on it. Yes, I actually use the same solvents ratio for the silica gel since it's the purpose of the thin layer one. When you test successful with your choice of solvents, you just carry it to the silica columns.
Yeah, I want to add to it, like the two solvent that work best for me is 19 chloroform and 1 methanol. And then the other one is 19 hexane with 1 ethanol. So as you can see here, the hexane and the chloroform are far away from each other. However, they had the same results. But when I used dichloromethane, it didn't work. So yeah.
So yeah, so the only way that you want to separate your compounds is try them out. I'm telling you, try them out. All right. Do you have any questions? I think I did answer that. Not 1-to-1. You can substitute the solvent to the use. Yeah. You can substitute it just to try. Since I've been substituting my solvents for like hundreds of times, I finally found my choice of solvents. But the downside of that is since that one gives you the best result, it doesn't mean that it's going to give you the best separations. So in order to from my point of view, in order to be success in this kind of project, you have to combine different combination. So for example, like mine, if you want to do isolate the compounds, I use petroleum ether and acetones, but it's only give me let me see, it's only give me like so for this slide, it's only give me separations in 1 the yellow here, 3 colorless, and then 2 of these like completely separated. But if I want to separate these below here, I have to try different solvents that can actually make them more separated. So it's just the point where you have to try like different solvents all over again. Show the picture of nightshade for compass. Okay, so for disclosure, yeah, we actually pick out the fruit only. But since Dr. Kavi, she had a sister that sister just plant plenty of this kind of plants in her garden. So I got that sort from that lady. With this one, I only saw it like a couple weeks, and now it's because it does not belong to a landscape, so they just take them off. It's so sad, but well, I did use some of the fruit.
So we have another question here. Where did you get turmeric root? From the farmer's market. Yeah, it's just any turmeric root, but it has to be fresh.
Yeah, you can get it from the market with the
Yeah,
with the plant that I have right here. I have to find them outside because they don't
yeah,
know that because they're unwanted. So they weed, so they invade your garden. So that people just take that off and without knowing that how it's gonna affect the cancer cells. Yeah, that's why I joined this research to like wake people up. Hey, this is really useful here. Just the different results. No, I don't think so because I've read multiple of the articles from this kind of plant. I actually have a list of the compounds within the plants that cure cancer. And they're from everywhere. They're from China. They're from Europe. They Africa, and so many in the U.S. They still have the same compounds within the plant.
yeah, same here. Thank you so much for visiting our booth.
Yeah, thank you guys. You're the best.
Yeah. If you have any question, please ask us and we will answer.
Yep, we're happy to
Yeah.
answer anything now. I'm glad that Wiswar decided to stay here to support us.
My CV just
Oh my God, which one? Do you like it that much?
yes.
Oh my
I was jumping from your presentation to Riasa's presentation. I was going in, going.
Oh,
I really want to visit her too.
Yeah.
yeah.
Oh my God, I'm so glad.
Yeah. I wish we were in different sections.
Oh, really?
I wish it's not. We were in the same section as Riyasha.
Oh, yeah, you're right.
Yeah.
Oh, this will count as either one field trip or one seminar.
I already have them. Oh.
I still missed one seminar, so this would be awesome. Turns out I still have a lot of pictures and video.
I used to hate that silica gel thing. Just to tell
I
you.
know.
Yeah, I remember when we did this.
And I still did not get the results. I was just like,
Yeah. Your sample is tough to work with.
yeah.
Yeah.
Hello. My sample tool. So yesterday I'm looking for videos to add to my slides here, because I think whenever I look at this poster, I don't have anything to say, like much to get attention. So I look back in my galleries and saw like tons of videos that I took. I'm so glad that I actually use it for this presentation. Yeah, so well, some of them have Dr. Carvey in it, and I don't know if I should put it up here or just leave it like that. Oh my god. What time is it? I don't even. 1 more hours.
Oh, you have 1 more hour?
Hopefully someone joins.
I'm like, by the time you were explaining, I'm just like, yeah, I did that. I just asked question for because I wanted to ask a question.
That's a good thing that you actually understand the inside of this project, because the reason why I put a lot of pictures and a lot of videos, because I know the feelings of looking or reading the articles that Dr. Carvey give me.
Yeah.
I didn't understand a thing.
Yep.
I don't even know what is. I didn't take OGO 1 yet back then, so I'm like, I didn't know what the heck is thin layer chromatography. What is the silica? What is the solvents? I don't even know. I can't. I mean, somehow I passed my Gen Chem class, but I don't really know how to identify which is solvent, which is solute, which is polarities. So I'm like, what the hell? But yeah, I mean, it's a good thing. I got a
Yeah.
lot from that research.
Is someone gonna continue? Like, are you gonna continue like the next semester too, or like?
Unfortunately, no.
What did you say here?
Unfortunately, no.
I was thinking about like joining that summer, because like what's the point of doing a research without like having results?
Yeah. I
Yeah.
wanted to continue mine, but I had 6 classes. I was like, and I started working too, so I'm just like, yeah.
Yeah, it's hard.
Yeah.
But yeah, I'm gonna ask him.
I'm glad that we have so many questions today.
Yeah, I was surprised by the questions that they were sending you guys. I'm like, they're actually interested.
I mean, I'm good. Because all of them pretty basic stuff. Because I know that not everybody into OGO 1 and 2. So they might have different questions regarding to the solvents, and apparently they did. Because I believe that during soft opening, when I first took the OGO Chem lab, we did the TLC online.
Wow. That's not even, yeah.
If I didn't join the research, I wouldn't know what the plate's gonna look like, because they basically give us a pictures with a dot and then identify which one is the non-polar, which one is polar, depending on the place that they put on that plate, instead of actually put into the solvent, let it run, or see it under the light. They just draw them out, and I'm like, oh my god, this is unfortunate for all the students that have to take that class without actually know how TOC works.
For me, when I took OChem 1, I did actually did the TLC on in the lab, and which was very like, it was like when I came to the research, I kind of knew how to do a TLC plate because of that. But right now I'm taking polymer, right? When I was taking inorganic, we transitioned to the online version, so I never got to do IR.
Oh.
And they, like you told me, like we were just given like IR spectra and we have to identify the compound, and now I actually have to work the IR machine. It's just like,
So for the IR, we actually didn't do it yet because we need the compounds that
compound, yeah, like the compound, and then you can.
they're gonna be just one band
Mhm.
in that fractions. So Dr. Chufan actually just pushed me to try. Oh, hey, put yours in the IR machine and see what functional group is at. And I'm like, but I'm not ready for that yet. But
Mhm.
actually, she was the one that introduced us to the second approach. So I'm so glad that I asked her, what should I do next? All right, just wait a couple more minutes for more students to get in. Just like 2 more minutes.
Hayden, if you want, we can start and then they can join us later.
Okay. So let me see. My name is Hayden, and I have my lab mate here, Afaf. We work under Dr. Jacob Derbati for this research that we extracting and isolating the anti-cancer components that found in the black nightshade leaves and the yellow turmeric roots. So for introduction, that we all know that there are several methods how to treat cancer, including the chemotherapy, biological therapy, radiations, but all of them always have side effects on the healthy cells. That's why we decided to test on the plants that already have the anti-tumor activities and the hepatoprotective activity within it. And that and usually the herbs have less effects on the healthy cells of the human body. So for the method, I use acetones to extract my black nightshade leaves. And in the first pictures right here is the thin layer chromatography, where I use this thin layer to identify which one is the best solvent to use to isolate the compounds later. And the outcome shows that the petroleum ether and acetones 4:1 actually was the best solvent to isolate, since I can see here that there are more than 7 different compounds within this plant. And then after that, after decided the solvents, we move on to silica columns chromatography, which is similar to the thin layer chromatography, but upside down. So we use gravity, polarities, and the solvents to isolate the compounds within the plant group. After that, we collect the fractions whenever the solution go down the columns. Then identify if we completely isolate the compound using a large TOC plate test on the fraction that we have collected. Since in one test tube, we have multiple compounds in it, we decided to try another approach, which is instead of the silica columns, we loaded directly the samples on the large TOC plate here. And then as you can see that there are multiple, like more than 10 bands that appear on the plate, which indicate there are different compounds, and we can easily scrape them out, put on a beakers, and then use a suction filtration to wash off the compounds down the flask, and then keep that flask in the safe place for us to test this later on. Okay, now Afaf can go ahead and explain a little bit about your samples.
Okay, so as Hayden mentioned earlier, our objective is to extract anti-cancer component from different herbs. So in order to treat cancer, both cancer cells and the stem cancer cells have to be killed. However, for chemotherapy, only cancer cells, it only kills cancer cells without killing the stem cells for cancer. So why stem cells, why cancer stem cells are hard to be killed? The reason for that is there a cytokinase called interferon 6. This protects the cancer cells from medication that is aiming to destroy their DNA. Also, interferon 6 sometimes convert the infection to stem cancer cells. So there is a research that found curcumin is able to kill both cancer cells and stem cancer cells. And so curcumin is found in yellow turmeric root, which is a herb. It's more safer to use than the chemotherapy and more effective. So for my sample, as you can see here, the first picture, this is the picture of my sample after extraction and evaporation. I used chloroform with methanol to separate each component from each other. So as you can see here, I only have 3 components. So the next step was separating this component in order to test them on the breast cancer cell. So in order to do that, I used the silica column. So for this method, it takes about 6 to 8 hours to get only 40 fraction. However, I collected 80 fraction, but I didn't see all the 3 components in there. Therefore, I moved to the next approach. The next method is I only used my sample directly on the silica plate. So as you can see here, I was able to see that 3 components clearly here. So each band represents 1 component. So after that, I scraped each band and then placed them in different beakers. After that, I got my components from washing off this silica by using acetone. So the next step we will do is that we will test each component on the cancer cell to see if they're effective or not.
Okay, So for the next couple of slides, I'm going to introduce you guys with details in each step that we did for the research. So first pictures here is the black nightshade plants that this is my samples that I use for my research. This kind of plant is like a weed-like plant, so they are everywhere. The pictures right here is I took on the walkway to STB building. This plant has been removed because they are not belong to the landscape of the school. But I did use some of the fruits on it for my research. And so whenever I collect the samples, I'm gonna weight it and then use acetones to extract the leaves right here using a blender. So we set up a blender, put the leaves inside, pull maybe 100 or 200 milliliter of acetones in it, blend it together, and the acetone gonna do the job. Like they're gonna be extract the components, the chemistry components within the plants. We put it on the stirring plate here, leave it for 24 hours under the fume hood. So we come back the next day, transfer it to the round bottom flask, you can see here, then plug it in the rotary evaporator in order to remove the acetones, remove the solvents out of the samples here. So after the acetone completely evaporated, the compounds is actually the yellow, the green bands right here. So they stick on the round bottom flask. So I'm gonna put some of the acetone to wash them a little bit, put them in the little beakers here, let it dry off by themselves, and I collect the gel like really concentrated blend roots here. So this is a little bit the video that I took whenever I done this. So you can see that the bubblings here. So the solvents actually collects here, so you can easily see it. The temperatures here is the pointing point of acetones in order for it to evaporate. Right, and then after that, after collect the plant brood, we decided to load it into the silica columns chromatography. So this is the columns that we loaded with the silica particles, and then the solutions here is the solvent of choice, which is the petroleum ether and acetone 4:1. And the next picture is here. Is the picture that I load my samples in the silica, and then whenever I turn the knob here, using gravities and difference polarities, the compound is going to run down here, and we collect the fractions whenever it's run down. So it's one amount each fractions. So let me show you a little bit here. So this is time lapse that I use. You can see that the sky behind it. This took 6 hours for me to complete. Before, the plan is only to collect 40 of the fractions, but when I complete 40 fractions, there's still colors in this silica in this column, so I don't want to waste it. So I just keep collecting until the column is white. So I end up with 112 fractions in total. But then, that's a good thing, since we can utilize fully of the samples that I had. Let me see. So then this is the pictures of the fraction that I collected. So this is like one amount of each, and then they have different colors. They might indicate 1 or 2 compound within it, but that's okay, since we can use the same thing to isolate them more and more. Right. So then after this, we collect the fractions, right? And then we test this on the large TOC to identify if they are if they are similar in the polarities. So whenever we see anything that similar in polarity, we just combine them. So we reduce the fraction, the number of fractions down to 16 instead of 112. So these here is really concentrated, and I mean, this is a really good result that I had. So for this method, after I combine them and I test it again to see if it's isolated for just one compound. So it's supposed to show one band on the plate. And then just couple of the fraction that show one band. Many of them show like maybe 2 or 3, maybe 4, 6. So we decided to move on with another approach, but we didn't throw it away, just keep it for next times. So the next, the different method that we try is instead of the silica columns, we load it directly on the silica, on the TOC plate, the big one, and let it sit in the chambers with same solvents for it to run up here. And this picture, it does result when it's run all the way here, when the solvents run is over here. So you can see here is the yellow band is completely isolated. If you use a UV light, you can see 3 colorless bands in the middle right here. And then this is completely isolated. The second black band is also completely isolated from the below. So the next step is I use the flat spatula, scrape out any bands that I see that is isolated, and then put it in bigger, right? So this silica actually contains the compounds that already isolated. The next step of this, I'm gonna wash this with acetones using a suction filtration. The silica is gonna stay on the funnel, and then the filtrate down will have my isolated compound. So this is a video that I took and how it's run. At first, it looks like it's overlap, but when you let it run, I mean, I just wish that we have a bigger chamber, longer blade. You can see it's isolate more clearly if it's run, because since the different in polarities, they're gonna run differently like this. And this process took around, I think it took around 3 hours to run up the solvents right here, the solvent lines right here. But when I recorded this time, my phone's died, so I have to stop it. But this clip gonna give you like an idea like how we did it. So next slide. Afaf's gonna continue introduce us. So we have the same techniques, just different samples. So it should be the same thing. All right.
Okay, so this is a summary of the steps I followed. So for the first step is extraction. So for yellow turmeric root, I separated the skin from the pulp. So as you can see here, I have 2 beakers, one for skin and one for the pulp. After 24 hours, I did the evaporation, which is the concentration. And the third step is to find the right solvent, the one that is able to separate separate the 3 components from each other. So for step 4, the first method I did is using the silica gel. So for this method, it takes time. It took 6 to 8 hours to collect the 40 fraction. However, I collected 80 fraction and I only was able to see 2 components. Therefore, I moved to the next method, which is only using my sample directly on a silica plate, and then using my solvent to see the 3 bands. After that, I script each band, which represent, so one band represent one component. So from there, I used acetone to wash off my component from the silica. So the next step we will do is we will test this component on cancer cell, breast cancer cell, to see if they are effective or not.
All right, thank you so much. My last slide here is just to give you a little bit more info about how we choose our solvents. So the tops here is the polar solvents, and then when it's run down, it's really non-polar. So in order to choose the solvent for your compounds, you have to play with its chart. You know, like how they are polars and non-polar comparing to each other. You just mix them with different combinations, or with one combination but different ratio. And it's going to give you a much more understanding how the polarities work on those compounds. All right. So yes, this is gonna be my poster. Thank you so much for coming. And if you have any questions, just
Thank you.
Okay, yes, I have questions. Is it possible to prevent yourself from getting cancer if you drink enough turmeric?
Actually, I found an article about that. So for aging, mostly for people in India, so they use turmeric a lot in their dishes. Therefore, they have less. So people there have less cancer. So therefore, yeah, it worked. It worked.
Okay, and for deadly nightshade, I mean, at first, I thought deadly nightshade was like a poisonous plant.
oh, is this actually a different type of that? This is the black nightshade one. It's different from the one that you mentioned. So they look similar, but they are totally different in the way that they shape the fruit. So you can see here, there are individual. I mean, like they have like a whole stem with multiple fruits on it. With the one that poisoned is just one in one stem. So this is this. So I can say that they have a poison, but it's not to the point that it actually can poison you. So we can avoid that that chemicals by extracting different various of the compound within the plant.
Okay, and what would be like the future purposes for extracting the essence of these plants? Like, would you maybe sell it to a company and have them distribute it as a vaccine or anything?
So this kind of research actually be done in multiple labs all over the world already. That's why we kind of like have an idea of how to extract them through their articles. But yes, so they extract the compounds, then they have the structure of the compound that actually cured cancer already. So we basically just follow their steps to see if we can have a faster approach, or we can basically this the similar thing that they did in the lab to isolate the components within the plant. We do have a Word document that have all of the compounds from this plant.
So to answer your question, there is an article I read about that. They use it as a supplement, like in pills,
Yeah.
instead of like vaccination.
Right. So this is more than like a pill to use.
Yeah.
Okay.
Mhm.
Thank you for your time.
You're welcome.
Thank you so much.
Thank you for stopping by. Poor Fab.
I want to see my cat. Wait.
What?
My cat is calling.
Oops. I don't have any support here. I only have my crocodile.
Well, like, I'm surprised they have questions. It's good.
To recognize, I mean, there are several students that actually presented that can be this question. They were in the oral presentation.
Oh, yeah.
God, I'm getting so hot.
I'm thirsty from talking.
Oh yeah, you're right. $20 more minutes. Maybe one more round. Let me add I have the possible compounds within the leads. I really want to show this to them, but I don't know how.
It's fine. Keep it short.
Oops. I don't know if I can put it here.
Hayden.
Yes.
So when I tested my components again, like each band when I test them. So for the first band, it's only one. It was only one.
Right.
For the second band, I didn't see anything.
The second band. What do you mean? How do you know it's second when you?
No, like so when I did the whole sample, there was like 1, 2, 3, right? The top and middle, and then the last one. For the top, when I test this one, it was only one band. When I did the large silica plate again. But for the middle one, I didn't see anything.
You didn't see anything even under the UV light. There's still not.
Yeah.
I don't know. Maybe
But like
some of the compounds the amount within that sample is too little.
oh
So for example, like I tell you on my slides that when I have a yellow, right? And then I have a gray one, but within those two, they have 3 more. Was that when I scrape out and then I test it again with the thin layer one, is actually didn't show, even though that it show on the UV light.
yeah.
Hello. Hey.
Hi.
How are you?
Good. So how's it going?
It's pretty.
Yeah.
Yes, it's actually fun. We have
Yeah,
questions and students visiting.
Okay, so
yeah, we had a lot of visitors.
oh, good, good. How many visitors? More than 10?
Yeah, that's like there's a couple waves. So I would say like around like 30 people.
Oh,
Yeah.
okay. That's cool. Yeah.
Yeah, and we had lots of questions.
Oh, okay. So what are the main questions?
So they just asked about the solvent choices, like how we chose our solvent to make it run differently.
Mhm.
And then some of them have a question about like how so what is the purpose of this? So after extract and then test on the cells, so what? So I'm gonna sell that compound to companies to make drugs or just that. And then
Okay.
it was like because this type of research has been done everywhere, not just us. So they use the compounds that I extract, put in the pill, and they sell it.
Okay. So can I, so right now I'm seeing, I think, slide number 12. Can I see from the top? So can you explain to me? You've been,
Oh yes.
yeah.
Okay. Let me see. Yeah. Let me present in the beginning.
Mhm.
So today, I gonna talk about the extracting and isolating anti-cancer components that found in the plate nightshade leaves and also yellow turmeric roots.
Mhm.
So we all know that to treat cancer cells, we have multiple methods such as chemotherapy, radiation, biological therapies, but most of them they can eliminate the cancer cell, yes, but they still have like a side effects on the healthy cells as well. That's why we turned it to a plan that already have the anti-tumor activities as well, the hepatoprotective activity within that plant, and for example, like the turmeric root. So for extracting method, we use acetones as a solvent to extract the compounds within the plant. And then when I finish extracting that, I test this on the thin layer chromatography to pick the solvents that can give the best separations within my compounds. And the outcome proved that the petroleum ether and acetones ratio 4:1 was the better solvents than the previous report, which is the ethyl acetates and hexanes for the plate, because you can see that on the plate they actually show more than 7 different compounds, comparing to the previous one is only 3. So after choosing the solvents, we move on with the silica columns chromatography, similar to TOC, but upside down. So we use the different end polarities, the solvents that we chose, and then gravity to collect the fractions whenever the compounds run down the silica columns. But when we're doing that, each fractions, they have multiple compounds in just one fractions. We successfully reduce the amount of compounds in one fraction, but there's still multiple in it. That's why we try to move to a different approach, which is we directly load the samples on the large TOC plate and let it run up in the chamber that fill with the chosen solvents. So after that, after the solvents run up to the top, we just use the flat spatula to scrape that off. And then identify them, label them with number and colors, just in case that we go back and see it again to identify which one is which. So for the next samples, Afaf's gonna explain a little bit more about her sample.
Okay. So as Hayden mentioned earlier, our objective is to extract the anti-cancer components from different kinds of herbs. So there is a study shows that in order to treat cancer, both cancer cells and cancer stem cells have to be killed. However, for the chemotherapy, it only kills the cancer cells without killing the cancer stem cells. So why cancer stem cells are tough to be killed? Because there's a cytokinase called interleukin 6. So this protects the cancer stem cells from different medications. So for example, if the medication is aiming to destroy the DNA of cancer cells, this protected. So therefore, there's a study shows that curcumin is able to kill both cancer cells and cancer stem cells. So curcumin is a component of yellow turmeric. Therefore, I use the yellow turmeric in this research. It's safer and it's more effective. So for the first picture, so this picture of my sample after extraction and evaporating. So I used 19 chloroform and 1 methanol to get this separation. So as you can see here, I only have 3 components in my sample. So after that, I did the silica column. So the problem with silica column is that it takes between 6 to 8 hours to get 40 fraction. Also, I collected 80 fraction and I still didn't see the 3 components in there. Therefore, I moved to the second approach, which is using my sample directly on the large silica plate. So after that, as you can see here, I get the 3 bands, and then I scrape each band and then store them in different beakers. And then to get the components, I use the acetone to wash off the component from the silica. And then the step after that, we will test the component on the breast cancer cell to see if they are effective or not.
All right, thank you so much, Afaf. So now I'm going to talk a little bit more in details about the step that we did in the lab. So this, I want to introduce this kind of plants is the one that that I use as my samples. This is called a black nightshade plant. It's like a wheat light plant, so it's grow everywhere. So this picture I actually took from my way to SDB. It's actually in a walkway, but I think they removed it since they redo the landscapes in front of the building. But after collect these samples, we waited on this balance like this, and then we use acetone in a blender to plant it in a blender, right? And then transfer it to a bakers like this, put a magnetic stirrers underneath, and then let it stir for 24 hours under the fume hood for the acetone to start extracting the components. After a day, I come back to the lab and transfer it to the round bottom flask and attach it with the rotary evaporator to remove the rest of the acetones out the samples. And after remove the samples, the solvents completely removed, I transfer it to a small beaker like this. Is actually have a gel looking plant group that indicate that it's really concentrated. So this is a video that I did took on when I do the concentration. So this is the rotary evaporator. The solvent's gonna come down here, and then we just collect that solvent and throw it away. Let's see. So after we have the sample, right? We set up a columns with silica and loaded with the chosen solvents, which is the petroleum ether and acetones 421. So the next pictures here is when I load all of my samples in this and then waited. And then when I turn this knob right here, the different impolarities and gravities will pull all of the solvents down here. And then anything that's non-polar is gonna go down first. Anything that's follower is gonna be a little bit more polar. This whole process take around 6 hours to complete. But it's a really fun project, so I really enjoy doing this. So this video is the time lapse video, so you can see it's really fast right here, but by looking at the sky behind it, it's like 6 hour long, so you can see like very clearly. So at first, I planned just to collect 40 of them, 40 test tubes, but turns out whenever I finished 40 test tubes, it still have colors in it. So I just keep collecting until though silica is totally white. But then so because of that, I ended up with 112 test tubes. And then after that, collect the fraction collection like that. I tested on the TOC plate to combine anything that have the same RF values. So you can see right here, they actually showing multiple fractions that's similar in colors, and then some of them are totally different in colors, which indicate that these fractions are somehow different in the components that it had in it. And then this is the large silica columns here. I mean, silica plate here. So when I see something that have similar polarities or IF values, I just combine them. So starting at 112 different fractions, I reduce this into 16. And this pictures here is the 16 different fractions. Some of them are isolated. For example, like this yellow fractions here is completely isolated from others. Some of them have 2 compounds within it, some of them 3, 4, 6. So we have to continue the same method. But since the columns takes a lot of time, so we decided to move on with another approach. These fractions, we still keep it. We don't throw anything out just in case that we have to come back and test it again. So different methods would be the large thin layer chromatography. So we load directly the samples on this plate, put it on the chamber, let it run with the same solvent. But when it's run to the top here, you can see that very clearly that the yellow band here is totally nonpolar. That's why it's run really fast up top. But since it's isolated completely from other components, we can use the spatula to scratch off the yellow bands here. And if you watch under the UV light, you can see 3 more isolated bands in the middle of these 2. So I was be able to isolate those bands as well as this gray band here, and then the second gray band here. These band is not really isolated, but I can use the same method to continue isolate them. So this method took around 3 hours to be complete, but it's faster than just the silica itself. And this is the video when it's run up to the top of the plate. At first, it looks like it's overlapped, but if you let it run, it's actually separated pretty good. So when it's run up top here, I scratch them off, put it in the beakers, and then filter it with the suction filtration. So the silica gonna be remained on the funnel, but the filtrate now will have my compounds. Let's see. So so now Afaf can talk a little bit about her yellow turmeric. We did the same techniques, but different solvents. All right, go ahead.
Okay, so this is a summary of the steps I followed. So for the first step, I did extraction. So my sample is yellow turmeric. So I separated the skin from the pulp. So after 24 hours, I did the evaporation, which is the concentration. And then the third step is to find the right solvent that is able to separate each component from the rest. So as you can see here, I have 3 components. After that, I performed the silica column to separate each component. So the problem with this method is that it takes time. Also, it's more expensive than the second method. Therefore, I moved to the second method, which is only using my sample on a large silica plate. So as you can see here, there is 3 bands. Each band presents 1 component. So from there, I had the 3 beakers that has 3 components. And then I used a vacuum filtration with acetone to wash off the component from the silica.
All right. Thank you so much, Afaf. And then the couple of last slides here. I just want to point out that how we choose our solvents for the isolating method. So as you can see here is the list of the solvents that lists from very polar, and then down here is a non-polar. So in order to choose the solvent, we just have to play with it. We have like a different like a backbone to choose the solvents here, but I did most of the combinations. I did 2 solvents at a time, 3, and then 4 solvents at a time, and then different ratios of those combinations as well, just to see that which one gonna be the best in separations of my compounds. So here is a list of the possible compounds within the black nightshade plant. The first one is the galactotigonin. This one is the most popular one. They actually have the anti-cancer activity within this compound, and these are some compounds that go along with it. If you're interested, I can send you a little bit of this info through email. Just inbox me right now and I can send it to you. All right, thank you so much for listening. There's all of my presentations. Thank you for coming.
Thank you so much. It's a great presentation, great work.
Yeah.
Good to see. So it looks like your the TLC method is better and faster than the column method.
Yeah, it's faster, but to be honest, I still can find some flaws within this method. So you can, let me see if I can show that again. So for example, like this beginnings, right? When I load my samples on the plate, you can see that since I use acetone to dilute the extract.
Okay.
When I drop them on the plate, it's actually washed off, so it's expandable.
Oh, it expands, yeah, because the amount, it's too much concentration there.
Yeah, so because of this, when it's run, it's actually run 2 different bands. So it's just like you load the sample cricket.
Okay.
Yeah, so when it's run up, it looks like mine looks like it's have two different compounds, but no, but it's mine just one.
One
And
compound.
and then when it's get past each other, it's overlap. It's just giving up and then overlap and overlap. So that's why the problems here is when I do this, right, I scratch off these.
Oh, so those two gray ones are same. You are saying that those are same.
They might be, yeah.
They might be,
That's the
right, yeah.
problem. So then I actually tried a different method, but I didn't have a chance to put it here because I didn't take any picture yet. I actually test the same thing, but with the one that I isolated from the silica columns.
Mhm.
So this actually eliminates the amount of compounds that when I load on the plate, it's already isolated a little bit.
Isolated, okay.
Yes. So instead of 13 different compounds from the original sample, this gonna give me samples only have like 3 or 4 compounds in it, and it's
Okay.
much easier than isolate than
Okay.
the
So,
original.
okay, if you can you go to the previous one?
Yes.
That you'll see. So how long it takes to run from top to bottom, the timing? Is it like 3 hours?
Yeah, it's 2.5, I think. It's 2.5.
Oh, okay.
Yeah, it's a long time.
So I was wondering, can you put multiple TLCs in one plate? Because one TLC tank. So that way, we can actually speed up. Instead of loading that much on one plate, you can use multiple plates, but you can put in the same tank, so it will run together.
Right. Yes, I actually did it once.
Okay.
But that time it wasn't a really good time for me to try it because I don't know why. The school decided to close soon. So I just put it on the chamber and then the security just like, oh, you have to get out. We closed.
Oh, okay.
Like, oh my God, I can't do that because if I do that multiple times, I lost my compounds. I lost my samples.
No, multiple times, multiple TLC plate in the same tank.
Right. Yeah. So that's why I'm kind of like, I got scared.
Oh, you got scared. Okay, I
So
see.
many plates.
I know. Yeah, that's a problem. Yeah.
Yeah.
But that doesn't happen.
Right. It's not happening like all the time. But I'm trying to like to do it since I don't have much of the solvents
I see.
left. So I kind of like, yes, I'm on the way to try to put multiple plate within a chamber.
I see. Okay, yeah, that probably because I see that because when you try to load too much on the TLC plate, you don't get good suppression. Because then, but one thing I expect, it will be because in your case you're getting 2 distinct bands here. You see in the gray one.
Yes.
If it is, too much material there, so you should get a continuous band instead of like a 2 distinct band. You see.
Mm.
I mean, so that suggests me that it probably 2 different compounds.
I see.
Because if it is a problem is you're loading too much, then the band has to be continuous too. But you're getting 2 distinct bands here.
Right.
Tells me it may not be, it may be 2 different compounds, not just that loading issue.
Yeah, it might be that too. But when I take it out and I test this individually, it still gives me multiple bands with
it still gives you multiple bands. Okay.
So,
okay. So that means they are 2 different compounds then.
Right. Yeah. So the one that I showed you earlier in the lab is actually from one band, but this time, since I have so little samples, since it's only one band, it's actually show a very distinct bands. So I believe that I can actually extract that, scratch that out, and then have a isolated compounds, I think.
So right now you combine those two, the gray bands, or they are two different
It's still the two different bands.
bands. Okay.
Yes. Since it's
But-
different, I don't want to combine them.
Oh yeah, because you don't need to
before
combine because you already did the big job. Then why do I need to combine? Okay. So I would suggest maybe one day we had to run it on an HPLC and see they're identical or are they different. Can we get a signal there? Stuff like that.
Yeah. I'm planning to try that, the XPLC, or at least run an IR on the one that I already isolated.
Okay, yeah. So which solvent that dissolves?
I use acetone to dissolve the.
Okay, so is that your sample dissolves in methanol too? Can you check that one?
Well, my sample actually dissolve in everything,
Everything. Okay.
but for testing for TLC is not. I don't think I tested with methanol before.
Huh?
And it's off. It's not show any band. It's just like a wiggling line going up.
Oh, okay.
Yeah.
Oh, so the methanol that you used as a TLC solvent or just for dissolving the compound?
For the TLC solvents.
Oh, TLC. Oh, yeah, that makes sense because it's a very polar solvent. Maybe it just goes very quick.
Yeah. Since I know for sure that my compound have a lot of polar components. If I use only methanol, everything just go up.
Yeah.
So that's why I think that's the reason why I give a little bit wicking looking whenever I do it.
Okay, yeah. So okay, so if it is if they can dissolve in methanol, we can run it in HPLC and see how that looks.
Cool. Yeah.
Okay, so we'll do maybe sometime next week or week after next. So we can try that.
Yes. Yeah. We definitely can try it.
Okay, very good. I like the your video recording of that TLC plates and the column. So how long how did you use your phone or how did you record that one?
Yeah, it's if you're using iPhone, then it's actually have that functions. It's called time lapse. And then it's just keep recording, but it speed up the process. So I put my phones there for like 3 hours, but the video come out just maybe 10 or 30 seconds. But
so how big is the video file then? It's a small file size?
It's not really big, but it's not small.
Okay.
It's in the quality of the images is pretty sharp. So it's short, but I don't think it's like it's less than a gig.
Oh, okay.
Yeah, but this is pretty good though. Like I don't want to film it for like 3 hours.
I know, yeah.
I just want to like speed them up, but after 3 hours, there's some day that my phone does not have that much of battery, so I have to stop it because it's like 3 hours of recording.
3 hours of recording, yeah. Okay, great. I'm very glad that actually.
Yeah, because I know that the students can't imagine that how it's gonna look. Just by looking.
Exactly. Yeah, it's hard to understand how this works. So if you have a video like that, it's very
Yeah, because this is how I learn. I have to record it. I can't just look at it.
Yeah.
Understand it. Yeah.
Very good.
All right.
Okay, thank you so much. Okay, good luck.
Thank you for stopping by, Dr.
Thank you.
Bye-bye.
Are we done?
Okay. Yeah. Let's go.
Oh, we did so good.
I can't believe we're done.
Yeah, I think it's 2:11, so we should be good.
Okay, yeah. I'm gonna go visit my friend booth.
Oh, they're in section 2?
Mm.
Oh, cool. Well, I don't think they have any friends.
Yeah. Bye.